नेटवर्क फार्माकोलॉजी, पता नहीं कितने लोग समझते हैं सब चीज़ों को,
नहीं समझ।
लेकिन ये आज के दिन में नीड है, मतलब टुडेज नीड इज नेटवर्क फार्माकोलॉजी फॉर एनी आयुर्वेदिक फॉर्मुलेशन. So I want to discuss some of the things because they have given only 5 to 10 minutes for me. So actually we know all the things, but how to interpret all the things in a scientific way. This is the need of the hour of today's research. Actually we know that our body is a very complex system. How our medicines work in our body. Actually, actually different toxicity study, pharmacological study, pharmacodynamic, pharmacokinetic. But nowadays there is a study named is bioinformatics study, which you can know and you can interpret all the Ayurvedic data in a better way. First of all, actually, I want to share some of the things.
This slide.
You know that different nidanas that they are for any disease to create in our body. If you divide our body mainly Koshtha, Shakha and Marmasti Sandhi. So for example, we take a medicine name is Arogavardhini. Whether this medicine is effective in Koshtha or it is Shakha or it is in Marmasti Sandhi, we can use the data like bioinformatics data. You can do all the things in your laptop or computer in your house, and you can detect in which way the Arogavardhini body is effective, and in which proteins, which amino acids, which pathway, which genetic, which are the genes. So these things you can detect in your computer also. So basically, what happens? How we will do it? The thing is that there are different websites are there, because first of all you should know whether this medicine is effective in what type of protein, what type of pathway, what type of genetic pathway. There are different pathways are there. Like in Ayurveda, there are 3 things we know all the things. There is Kshiradati Nyaya, Khalekopata Nyaya and Kedarkulya Nyaya. Our medicine also asks all the things like this Khalekopata, Kedarkulya and Kshiradati Nyaya. Similarly, in modern science also they told that the medicines are working as either it is metabolic pathway, genetic pathway or signaling pathway. The similar things. What they told and our ancient literature are told, these things are similar. And we can detect all the things by this Ayurvedic, this bioinformatics study. So basically what is network pharmacology? Network pharmacology shows the interaction between the bioactive compounds and the pathways related to any disease. In Ayurveda also there are different dosha samasa, because for example we are suffering from coryza, for example nasal discharge is there, different nidanas there. It may be aharas nidan, it may be viharas nidan. Similarly also we can detect in which nidana this coryza will be appear in our body. So the things by which you can know is this network pharmacology. So network pharmacology is between the drug and the disease genes. We can establish a relation between the drug. For example, you take Lakshmi Vilas Ras. And how this Lakshmi Vilas Ras, what are the specific genes in which the Lakshmi Vilas Ras, because in Ayurveda they told that Purusham Viksha. Every medicine does not work in every patient. So in which patient this our Lakshmi Vilas Ras is effective, which Purush, which Prakriti people this is more effective, we can dictate by also this network pharmacology. So this is a new drug designing program, and also this drug target, where the drug will be effective, the target area, the pathways, all the things you can know by this network pharmacology. Basically, if you know all the things, we can interpret our Ayurvedic medicines in a better way. For example, we have done some work in our laboratory. For example, actually I have taken medicine known as this Kravyad Ras and this Snuhi Dub. Actually, in Ayurveda, they have mentioned that this is effective in case of leukemia. We have taken that leukemia, whether this Snuhi has some effect in leukemia. So for that, actually, we take this bioinformatics study, and we find that it is more lesser the proteins and the amino acids, the pathways are more in the prostate cancer, because in Ayurveda they told that it is effective in different type of arbuda or different type of granthi or different type of sotha or gulma. But when we go for the bioinformatics data, we find that this Snuhi is more effective in case of this prostate cancer. So these are all the data that there. But it is not showing in this screen also. Actually, now another example, we take this Krabhyadras. There is a formula in Ayurveda which is Ajirna Adhikar. We find that mainly where it is effective, the Krabhyadras. What we find that mostly in Renian angiostensin system, it is more effective. For example, because in Ayurveda there is in Phalashruti of a medicine, there are many more diseases are there. In Ayurveda also, in Krabhyadras, they have described it is in Jalodhara. It will be effective in Jalodhara. And also we find that, I do not think how many of our Vaidyas are using this Krabhyadras Jalodhara, but from my bioinformative data, what we have detected that it is more effective in Jalodhara, because it is the amino acids, the proteins, the pathways, the genes are mostly which are having this Jalodhara, it is more effective. So in this way you can detect many of the things. For example, we have taken there is different pathways are there, like cake pathway, genetic enrichment pathway, geo pathway. Similarly, in Ayurveda, what we told that this Khiradati Nyaya, Kalekapota Nyaya, Kedarkulya Nyaya, similarly we can detect in which Nyaya it will be effective. So these are all the things you can determine by this network pharmacology. Also, actually multiple studies are, because in Ayurveda, in every medicine, actually we are having more than 3,000 molecules in every plant. For example, we take Snuhi. After doing the LCMS study, we find that more than 3,000 molecules are there. So from how many molecules are effective in this prostate cancer, and how many molecules are effective in this leukemia, we can detect by this network pharmacology data. So this is a method. Actually, there are two type of method. One is called a dry lab method, and another is called as wet lab method. Dry lab method comes when the molecules which are described already in your different books, and we can take that molecules and just different websites, we can detect in which way it will be effective. And this wet lab method, where we have to detect the molecules present in your medicine. For example, you take Snuhi, we can detect whether what are the molecules present, because you can go for this LCMS study, that is liquid chromatography mass spectroscopy, and also for any compound, that's GCMS, any type of compound, you have to detect the compounds first. Then only you can go further. So actually there are different websites are there by which you can detect. First you should go for the PubChem website. PubChem website you can download the chemicals first, the structure of that chemical, the structure of that molecule. Then you can go for the Swiss ADME. First you have to detect because more than 3,000 molecules you have to detect what are the molecules present in my medicine, which are more effective in case of prostate cancer. Then you have to one by one, you go for the IDMA data, that is you can go for the Swiss ADME. This is a site called Swiss ADME, where we can get the bioavailability of that molecule, the drug likeness of that molecule, different physiochemical data and different pharmacokinetic, pharmacodynamic data of that molecule in this site, this Swiss ADME site. This is the first step. Then you can go for the toxicity data, whether that molecule have any toxicity or not. So you have to go for this. There is a site called as drug likeness site. There is a called as predict toxicity profile. For example, there are different types sites are there. They can detect whether this molecule have any immunotoxic effect or any like carcinogenic effect or hepatotoxic effect. You should put that molecule in that site and they will told whether these are molecules. So you have to exclude all the molecules one by one. Then you can go for this binding DB database where you can detect the proteins, the amino acids. So all the things you can know. Actually, time is very less, but this is a very good techniques for our Ayurvedic medicines. We should go for all the things in a better way to establish our facts whether this medicine is effective or not in a because for clinical study or experimental study or any other type of study में बहुत पैसा भी खर्च होता है और हमें वो डाटा नहीं मिल पाता. So I wish all of you that you should go this bioinformatics study. Thank you everyone.